as empiricists or natural historians, like many crabbers are, anytime we're drawing some inference or we're extrapolating or making a prediction about something that's going to happen in a change, we are making those assumptions or making those inferences, those predictions, those extrapolations based on a model of the world, how we think the world works, regardless of whether you're a theoretician or a natural historian, right? When we model, and Andres is modeling this system to understand what otters are going to do, he's doing the same thing that crabbers are doing when they're extrapolating to what they think otters are going to do. So models underlie the way that we, as humans, as scientists, but as humans, make inferences about the real world. The problem with that way of doing science, of knowing the world, the fact that we are using models to understand the world, is that our models are not always right. Even the correct model can be wrong in a different context. So I want to show you that with this example. So many of you know kelp forests for the so-called alternative stable states they exhibit. They can either be in healthy, vibrant, species-rich kelp forest states, or what we consider less desirable, depauperate urchin barrens, right? Kelp forests are kind of a classic example of alternative stable states. And on the left-hand side here, oops, is that going to work? No. Where's my pointer? There's my pointer. There it is. Nope, it's not showing up for you guys the same way it's showing up for me. These lines here, represent a couple of time series from sites from Southern California, San Nicolas Island. It's a 40-year time series going from the top to the bottom. And what these time series exhibit are these classic alternative stable states. So a site can either be in a forested kelp forest algal-rich state, or it flips over to be an urchin barren. So over the 40-year time series here, you see these classic alternative stable state dynamics, where the system is either in one state or the other. Formally, you can describe that as with a system potential. So these two wells are alternative basins of attraction, alternative stable states. And that way of modeling kelp forests, of describing kelp forests as exhibiting these alternative stable states, works for a bunch of other sites too. So here, going from left to right, you can see these basins of attraction, right, at many of these sites. But it doesn't work for all sites. As Zach showed, at sites that have a lot of substrate rugosity, really high complexity, you don't get alternative stable states. You get kelp forests that have a high abundance of urchins and a high abundance of kelp. Our model of kelp forests doesn't work for all places, even around the site, let alone around the globe. So that's the problem. Model appropriateness may itself be context dependent. Now, there are exciting new tools, new methods, data-driven methods that give hope to many that we one day will be able to use these data-driven AI machine learning, scientific machine learning approaches to learn the equations that govern systems from the dynamics themselves. Okay. Another one of my students, Shan, is working on that, showing that you can, without positing any particular model, feed data into these algorithms, give it a certain number of variables, certain operators, and it can evolve to learn the equations that could describe those dynamics. It's been shown in principle with high-powered simulations and simple lab experiments that really works. It does work. But as Cheyenne's work is showing, and she'll present this later this year, is that the types of data, the types of attributes that ecological data would need for this to work universally just will never be attained. It's just impossible for ecologists to collect the types of time series that these methods still require. More fundamentally, although these methods have been proven to be really good at forecasting, they don't necessarily provide understanding, right? And that's what science is about, in my opinion at least. So everyone in the lab studies species interactions. We're all interested in the fabric of nature. But what the way my thinking has evolved over time is it starts from the point of all models are wrong. Every model. We might think it's correct, but it's a wrong. It's an approximation to reality. And so the real question is not which models are correct or even which models are best. But the real question is how much approximation can we get away with to still be able to answer the questions that we're interested in? How much approximation will allow us to explain or describe or predict? So as Bruce was saying, we've been pursuing these kinds of questions, these core questions, in lots of different empirical systems, in kelp forests and intertidal, in streams, and most recently also in plant pollinator systems in the Rockies. But what I really want to talk about there is the fundamental challenge of working with empirical systems and coming to a point where you feel like your models are an appropriate, useful approximation of the dynamics of the complexity of those empirical systems. So, like I said, a lot's happened since tenure. Our lab studies this core question at 3 different scales, right? Some of our work is kind of at the level of individuals, where we're interested in the differences between individuals, within species and across species, to ask how much of that variation, how much of that heterogeneity do we need to be able to approximate in order to understand higher order properties of systems. We also study complex food webs, whole networks of interactions and other forms of complexity to ask, what do those forms of complexity mean in terms of how we can represent complex systems and their dynamics through time? But most of my recent thinking, most of my recent work has been at this intermediate level where we're really focusing on modules, small groups of species, not whole systems, not individuals, but just smaller groups of individuals, 3 to 4 species, 2 to 4 species. And in fact, for today, we're going to ignore everything except for, we're going to assume there's a single predator and a single prey. All of the kind of things I want to talk about today are really most easily thought about when we avoid a lot of that complexity that we know is there. I just want to focus in on the complexity of pairwise species interactions. So I'm going to focus on just 3 papers that we've published in the last couple of years. And again, the context here is the ultimate goal is, are we interested in describing, predicting, or explaining systems, and how do we go about approximating real systems in order to do those 3 things? The key biological concept for today is the so-called function response. I'll talk about more in a couple slides, but what we're really trying to describe is the rates of interactions between species. So on the y-axis in this case is the feeding rate of a predator, how many prey it eats per time. This is of an individual predator. And on the x-axis in this case is the prey abundance. Right? In this case, the more prey there are, the higher the feeding rate, and it saturates at some point. Now, there are non-ecologists in the room. I want to make this talk maybe a little more interesting by highlighting the fact that the types of models I'm going to be talking about apply not just to ecology. So anytime you hear me say prey or resource, you might want to think substrate or reactant. Every time you hear me say predator or consumer, interpret that as enzyme or catalyst. When I say feeding rate, just think reaction rate, uptake rate. Right? The types of models I'm talking about apply across all levels of biological systems, from chemistry up to ecosystem scales. My outline for today is as following. How am I doing on time? Ooh, yeah. I'm going to talk about the linearity, the approximate linearity of these function responses. And so the first thing I want to talk about is a biological I guess a method I discovered, or a way of thinking about these systems that I realized allow biology to cause linearity in these systems, something I didn't appreciate before working on this project. Then I want to take us into the world of statistics for a little while and talk about the ways that our experiments, our data sets, and our ways of fitting models to data can bias us towards thinking systems are more nonlinear than they actually are. And at the very end, I want to distill the notion, or distill, is that the right word? Get rid of the notion that in order to understand the complexity of the world around us, the complex dynamics that we oftentimes see at play, that we need complex nonlinear models. I want to instead posit that we can obtain complex nonlinear dynamics from linear interactions. That is, we don't need complex nonlinear models to explain nature. Hopefully I'll get that all in the next 30 minutes. See. Okay, so as I said, the core component concept of this talk is the so-called function response. On the y-axis is the feeding rate of an individual predator. On the x-axis is the predator's prey, focal prey species. This concept of function responses is fundamental to ecology. Pretty much every intro to ecology course teaches about function responses because it applies not just to predator-prey, plants and their resources. As I said, it applies at many different scales of ecology. There have been thousands, literally thousands of functional response experiments conducted, many different motivations and reasons, but that plot on the bottom right there plots two parameters from a function response against each other, and every single one of these dots is a different experiment that's been conducted. And likewise, we've got dozens and dozens of different function response models that have been derived over the years to describe the complexity of function response models, of function response relationships between feeding rates and resource or prey abundances. I'll even point out that functional responses are so important that they lie at the heart of predicting climate change. So this paper here, published up a couple of years ago, pointed out that our uncertainty in how we describe zooplankton grazing rates is the largest source of uncertainty in our global climate change projections, right? Particularly for the marine carbon cycling, but it has a huge impact. How we describe just how a zooplankton feeds on a phytoplankton is an extremely most sensitive point in those models. So function responses have been around for a long, long time, models. They go back to Lotko and Volterra, 1903, I think it was, or maybe 1901, with the so-called, now it's referred to as the linear type 1 function response. Go back slide here. This straight line right here, okay? This line describes a system where the feeding rate of a predator increases in constant linear proportion to the availability of the prey. Now, in 1959, Holling came along and did some seminal work on function responses and coined the term type 1 and what I'll talk about later, type 2 function response. But he actually, when he defined the term type 1 function response, he actually drew a rectilinear model. It went up to a point and then cut across, two-part linear, because he, like most empirical ecologists, would say that, well, no predator can feed infinitely fast. But that terminology didn't stick around, and so we now refer to the type 1 function response and sometimes the rectilinear type 1. Now, as I just alluded to, empiricists hate the type 1 function response because it's just biologically unrealistic. No predator can feed infinitely fast. Right? Theoreticians, on the other hand, love it. It's the simplest possible model. You can do a lot of analytical work with it. It's the foundation of predator-prey ecology. Both camps, however, dislike the rectilinear functional response. Yes, it seems more realistic, but to quote someone, nature abhors corners, right? So it can't be a realistic mechanistic description of reality. The theoreticians don't like it because it's two-part, and so you can't do the math with it very easily. It's a really complicated math. No one has, to my knowledge, ever worked with a rectilinear model. Now, why would one think that a linear model could be good enough? Well, there could be two reasons. Good enough to describe a functional response of an empirical predator. There could be two reasons. The first might be that prey abundances just don't vary very much over space, over time, such that linear is actually a pretty good approximation over that range of prey abundances. Okay? Or feeding rates may just never be high enough. because prey abundances never get high enough. They are always in this range down here, so insufficient magnitude. I'll give you examples of both. The top right graph there is from my thesis work, actually, where I'm comparing linear estimates with nonlinear estimates for this particular prey species fed on by a little New Zealand whelk. Okay? What you can see is that these estimates are barely different from each other. Linear is just about as good as nonlinear for this particular system, despite the fact that whelks in experiments could have very nonlinear saturating function responses. They have handling times of hours to many days. They could saturate very quickly, but they don't. Likewise, Kyle Koblenz, some of you remember, I've been continuing to collaborate with him. We showed by looking at all those 2,000 or so functional response experiments that have been conducted in lab experiments, or in lab systems, and getting empirically relevant estimates of prey abundances to try to take those lab experiments out into the field. We then measured, well, at realistic prey abundances, how linear are functional responses? Are they down in the corner or are they up here in the saturated range? And the answer is, here's the saturation index, linear on the left-hand side, saturated on the right-hand side. The vast majority of functional responses at realistic prey abundances are linear. Okay, so two, what we might call side effects of biological variation, but also how we conduct experiments. But I was interested I wondered, is there a biological mechanism to also get linearity? The previous ones being just variation in nature or limited variation in prey abundances. I was curious, is there a biological mechanism that is intrinsic to the predator and the prey themselves rather than the variation in the system? To tell you what I ended up thinking about, here's the type 2 functional response. The type 2 functional response, predators have a handling time. They have this saturation. They, at low prey abundances, they're search rate limited. There's not enough prey around, so they bump into them relatively slowly, so the feeding rate is low. But as prey abundance increases, more and more of them are busy handling prey and are therefore not able to search for any more, and that's what limits their feeding rate. What I realized is that this type 2 model, as well as all other models that have been published, all dozens and dozens of them, make the assumption that a predator can only be either searching for prey or handling one prey. Right? Searching for prey or handling one prey. As soon as they're handling one prey, they stop searching. That's what limits further feeding rate. Kind of like this. So you're either in a searching predator pool or you're in a handling predator pool. So I asked the question, well, what happens when we allow individuals that are handling one prey to continue searching for another prey? Well, what you can see is that functional response came up a little bit. They're feeding faster, right? But it still saturates. Well, we can play this game even further and ask, what happens when we allow predators to search for an arbitrary number of prey? Right? They're able to handle 1, 2, 3, up to n possible prey species. What you can see is that we recover the rectilinear model, right, at the asymptote. Now think about all the species that model actually applies to, right? Otters can come up with an armful of snails. This puffin is a beautiful example, feeding on multiple prey at a time. Lots of species. Anything that digests multiple prey in its gut, fish, that's what their feeding rate is limited by, not their handling, true handling, but by digestion rate. They will exhibit this rectilinear functional response, are expected to. The point, however, here is that we have a region, an increased region over which we expect linear function responses. We've gotten this linear region, expanded this linear region of low prey densities that we didn't have before. So the natural question was, well, how many predators does this actually apply to? It could apply to a lot. Well, one of the things that we did is we went back to all those 2,000 function response models and fit this new model to the data, right? And asked, used maximum likelihood methods and asked, well, how many of the experiments actually are supported by all the different models that we have? What we see is that roughly a quarter of the time of the experiments are consistent with, not necessarily show exclusive support for, but are consistent with some degree of linearity in the functional response. And that this is true for across all different taxonomic groups that are in this giant database. So I've split these Venn diagrams here. There's two types of studies, ones in which the prey are not replaced and others in which they're replaced continually. And so I have to fit different likelihood models for those. But this darker outlined region are all the models that are consistent with some degree of linearity, roughly 25% of the data sets, of the experiments. Now, you might also already be thinking, well, what determines how many prey a given predator can feed on at a given time? How many can it handle at a given time? There's a paper out in the literature in the 1980s that had this prediction that the larger you are relative to your prey, probably the more things you can handle at a given time, right? An otter feeding on lots of little snails can feed on more snails at a time than an otter feeding on one giant urchin, for example. And we see evidence for that across all these data sets. So these, whether treating, excluding some of the data sets where one prey at a time is consistent or is not consistent, it doesn't matter. You see this positive relationship between the number of prey that a predator can feed on at a time, estimated number of prey that a predator can feed on at a time, as a function of the body mass ratio of the predator relative to the prey. Now, it's a really poor fit, right? There's not much explanatory power here, but we have learned something about what might be determining how linear, the probability that a predator exhibits a linear functional response. The graph on the bottom here is the same data, just showing you that it works for all different types of taxonomic groups, whether it's insects, crustaceans, fish, arachnids, or other types of consumers. So to me, the lesson from this first part of the talk is, as I said at the very beginning, all models, including the non-mathematical ones, including non-mathematical models, are approximations of nature, approximations of reality. And the real lesson for me from this first part of the talk was empiricists shouldn't dismiss mathematical models even when they seem biologically unreasonable. We might just be missing that one way of describing some aspect of biology that allows us to make sense of more biology. It's not, all the models are still wrong, but we might just be missing that one piece that makes models actually describe simpler systems than they seem to appear. I'll skip that slide for the interest of time. Okay, part two. I now want to dive into the world of statistics, which is what I've spent a lot of time on since getting tenure. It allows you some flexibility to think. I've tried to teach myself statistics. So I want to talk about how the ways that biology, statisticians, when they try to formally connect models to data, right? That's what statistics is. How the ways we've been using those statistics to draw inferences from our experiments, from our data sets, and how we design our experiments has been biasing our inferences regarding the complexity, the nonlinearity of predator-prey interactions. There's a couple parts to this section. And the way I want to frame it is in a different aspect of functional responses. So for some 40 years, there was a raging debate in the literature about the nature of functional responses and whether you needed to take into account not only the prey abundance, the prey's abundance, but also the predator's abundance. So at one camp, we have ecologists who consider functional responses to be prey dependent. That is, the feeding rate depends only on the prey's abundance. At the other end of a spectrum, you could call it, are those that say, no, it's not just the prey's abundance, little capital N, you also have to take into account the predator's abundance. That's because predators interact with each other as well, right? So if you have predators that are interfering with each other, wolves and hyenas, corals, whatever, right? When they're interfering with each other, that means they're wasting time taking time away from their ability to search. Right in the middle of those two extremes, the prey-dependent and the consumer-dependent ends of the spectrum is the so-called ratio-dependent camp. That is, it's not prey-dependent, it's not just prey and predator abundances that matter, but the ratio, a simplification, the ratio of prey to predators that matters. Like I said, there's been a raging debate. There's books published, there's the fallacies of ratio-dependent predation. Lots and lots of papers arguing back and forth. Unfortunately, much of this debate was done without data. These experiments are hard to conduct, as many debates in ecology are, right? But why this debate was so fierce is because it really matters whether you have a system that is determined by prey-dependent function responses or consumer or ratio-dependent function responses. That is to say, which worldview, so to speak, is right determines how you explain whether systems will cycle or whether they will just exhibit stable states, equilibria. It will determine how whole trophic levels or food chains respond to perturbations like nutrient enrichment. The one camp, if you add energy or nutrients at the base, the whole food web will grow. In the other, with the other style of model or way of writing these models, you'll get this alternating trophic chain, cascade of effects. It even matters how we interpret or how we think about the way that ecosystems function. It impacts the very nature of trophic pyramids. Under one camp, you expect the traditional biomass period, few little biomass at the top, lots of biomass at the other. In the other camp, you expect inverted biomass pyramids, which have been observed. But like I said, there was relatively little data. Over the decades, every decade or so, someone came along and synthesized all the papers that had been published and performed meta-analyses like this. This is an example from one of the key ones published in 2010, where they took one particular key function response model, the Arditi-Aksakaya model. The reason why they focused on this, it has one parameter, a little m, that determines the strength of predator dependence. So when little m is 0, you're in the prey-dependent end of the spectrum. And when m is 1, you're at exactly ratio-dependent, and anything larger means you have, or anything larger or lower, anything in between means general consumer-dependent. So that paper was published in 2010. I had a collaborator come over on a short sabbatical for half a year, and we started working on some projects. I had done some experiments thinking about predator-dependent function responses that I actually talked about during my tenure talk. We started collecting data sets for a different project and realized, well, we had almost twice as many data sets in our hands already as the previous publication. Why don't we go ahead and see what the distribution of these M interference strengths looks like? And this is what we saw. Very much like the previous analysis concluded as well, right? Very similar data. The average of all our estimates was very, very close to 1. So ratio dependent is the best model. Okay? But one of the things we realized is that those estimates are fundamentally biased. So here are the same estimates, but now arrayed, with each study on a different line here. And I've ordered them by decreasing sample size, how many replicates they had in the experiment. So a huge effort went into these experiments up here. I think 500 or so replicates. These had on the order of about 10 or 15 replicates. What you see is that the smaller the experiment, on average, the greater the estimate of interference strength. Our experimental design is nothing to do with biology. Our experimental design is determining our inference, or is controlling our inference about which of these models is the best. The fundamental cause is a statistical one. It's been known in the statistics world for a long time, but not among ecologists. If you have observations that are changing their time with some amount of noise, you get a distribution of observations. when you have a distribution of observations, anytime you fit a statistical model to them, you will get a distribution of estimates. Because our estimators, or our statistical estimators, are nonlinear, due to Jensen's equality, these parameter estimates will be biased. This is true even for linear mathematical models, like the type 1 function response model. When you put it in the context of statistical model, that statistical model makes that mathematically linear model a nonlinear model. So you will get a curved likelihood function. The less data you have, or the more noise and variation you have in your system, the more biased your estimates will be. And this applies to all function response parameters. It applies to all statistical models that we use in ecology. Right? So that decades-long debate has been fueled by this intrinsic statistical bias. We have been overestimating the so-called, the magnitude of the so-called true parameters, which has been causing us to infer that species interactions are more linear or more nonlinear than they actually are. Now that all supposes that we actually know the true model. What I just talked to you about was we assumed the Arditi-Aksakaya model. We assumed that we had the right model and we're simply drawing inferences based on the parameter value. But there's an even bigger, more fundamental problem related to aspects that I want to talk about next, in terms of when we try to use statistics to compare the performance of different models and asks which model is the best. Right? Say we have a suite of models and we're trying to ask which is the most consistent with our data, which is the best at describing and fitting our data. Well, the traditional way of doing that, no longer traditional actually, yes, I guess came to ecology in the early 2000s, is to use information criteria. Things like AIC or BIC have a couple different criteria that people use. What AIC and BIC and these information criteria try to do is basically implement in a quantitative way a comms razor. You're trying to balance a model's fit to the data with its complexity. Right? You want to keep your model as simple as possible, but no simpler, but still describe your data. And so AIC does that by estimating the likelihood of the model and then penalizing it by some complexity measure. In this case, it's just twice the number of parameters. The more parameters you have, the more complex your model, the more you penalize it. And you're trying to find that sweet spot, right, between a model that is not overly complex, not too simple, but the optimum right in the middle. I'll skip this slide too, yeah. So here are those same data sets that I described before, but now I'm using AIC as an example to applying it to every single data set and asking which model performs best based on AIC in describing those data. So this is a complex graph here. So across the rows are the different data sets, again with decreasing sample size from top to bottom. Every single little dot here with a different color represents a different model. In this case, we have, 8 different models that we're comparing. The gray outline here, anything in the gray box means the data isn't, there isn't enough data to distinguish models. Those models are all equally well performing. Okay, now that's a hard one to look at. What I want to show you is this way of looking at the same data. So on the far left-hand side of this graph, I'm looking at only those data sets that have sample sizes of 20 or less, right? So these data sets down here. And I'm going to increase the sample size from left to right, including more and more and more of our experiments. On the y-axis is the model that we infer to be the best at describing those data. That is, what proportion of the experiments with sample sizes less than 20 provide support for this model versus this model versus this model. What you can see is that as you increase the sample size of the data sets, as you include more and more data sets, our inference regarding which models are the most appropriate for describing ecology changes. right? So it's not just a parameter bias, it's a model inference, a model comparison problem bias that we have with these types of experiments, these sample sizes. And it gets even more complicated. It's not just that our indices are biased, our indices are too simplistic in how they describe the nonlinearity of different models. So as I said before, AIC and BIC and many other forms of information criteria quantify the complexity of model simply by the number of parameters. BIC does a little more and takes also into account the sample size of the experiment. However, what Daniel and I realized that is ecologists aren't recognizing, particularly in the functional response world, ecologists aren't recognizing that two models that have the same number of parameters can still fit the data into different degrees. That is, they could differ in their flexibility, their ability to accommodate the data. A simple example is just a linear function, right? Yx equals a plus bx, two parameters, versus a power relationship. Still has two parameters, same complexity, but clearly the power law relationship is more flexible. It can accommodate some simple nonlinearity. Well, that's true for many of these function response models as well. So we brought to the ecological literature this other more complicated information criterion. It's the Fisher information approximation. It too uses the likelihood of the model to estimate fit. It too has this measure of the parametric complexity, but then it also includes this measure of how flexible models are, having accounted for the parametric complexity. And importantly, that model flexibility is not just a function of the model, but of the data, of the experiment, how we design our experiments. So here's a classic functional response experiment. You look at the number of prey eaten as a function of the number of prey provided. If you allow the predator to feed on a maximum number of 20 prey, these blue stars, right? That's the maximum number of prey that you are setting up your experiment with, and you allow that the minimum number of prey that a predator must eat in an experiment to be 1, otherwise you discount it. Right? Then that yellow region defines the possible space of data. Okay, your models have to be will differ in the point I'm about to make is that models might differ in how much of that yellow space they can fill, even after you account for their parametric complexity. So here's an example. Here's the linear type 1 function response. This is a complicated figure. Oops, I overlaid the darkness on the x-axis here. On the x-axis here is the maximum prey abundance that you design your experiment with on a log scale, so from 20 to 200. You have a very small experiment, you have a very large experiment. Each of these lines corresponds to a different number of predators in your experiment, whether you have 1, 2, 3, 5, or 8 predators in a given treatment. On the y-axis is that model flexibility, it's geometric complexity, right? So you can see right off the bat that how you design your experiment will give you a different value, a different measure of how flexible that model is. This is even just for the type 1, where there isn't true flexibility, it's just moving up and down. But here's the important point. Different models have different amounts of flexibility. So this is another one parameter model. And I'm plotting here the difference between these two one parameter models, the ratio dependent model minus the type 1 function response model. What you can see is that the ratio dependent model, you're biased towards inferring that ratio dependent model. For all experimental designs, except when you have only a single predator, you are more likely to think that this more flexible model is fitting your data better, is performing better at describing your data. And if you had designed a different experiment, then that bias would be a little less, but you would still be biased towards thinking that your system is more nonlinear than it is. Unfortunately, it gets even more complicated because the bias itself can switch. So here are some 2-parameter models on the left axis, on the left column, and here are some 3-parameter models on the right. I'll point out this one here to highlight. So this is actually, I'll do this one here. This is the Halling type 2 that we've talked about. Here's a Hassel-Varley. So in blue, at small prey abundances or high predator abundances, you're biased towards the Hassel-Varley model. But if you were to conduct another experiment and use higher prey abundances or lower predator densities, you'd be biased towards the type 2. The models haven't changed. All that's changed is how you've designed your experiment, and you're going to be biased one way or the other in terms of which model you are more likely to infer is the correct model. And some of these differences can be huge. The standard difference that people use for AIC and BIC in terms of determining which model is far better than all others is 2 AIC units. For many of these comparisons, just the model flexibility itself can far exceed 2 units. This then explains the graph I showed you guys before, why changing sample sizes so drastically changes our inferences about which models are most important. Oh man. Okay, so that was section 2. Simpler models may be more appropriate, may approximate reality better than experiments actually imply because of the statistical biases. So in the last section, squeeze this in, I want to talk about the thought that many ecologists have that in order to explain the complexity of nature, all the complex dynamics that we see out there, you have to have nonlinear complex models. I'm going to go back to that first model that I talked about in the first section, this model where we have predators that are able to consume multiple prey at a time. Right? The model that gives us this expanded region of linearity at low prey abundances. So this is where we're going to do some math. Everything else before was data and statistics. We're going to go back to what is probably the canonical model of predator-prey ecology, the so-called paradox of enrichment model, the Rosenzweig-MacArthur model. We're going to do isocline analysis. Okay? Isocline analysis is a graphical way of inferring the dynamical properties of a model. So the way to read this graph here is this vertical line denotes the prey abundance at which a predator's population size is not going to change. It's at equilibrium. If you have prey at higher abundance, the predator is going to increase. If the prey are below, the predator is going to crash. Likewise, this parabola describes the predator's isocline. If the predator is above that isocline, then the prey will go down. If the predator population is low, then the prey will increase. Okay. If these two isoclines intersect to the right of this maximum, in fact, if they intersect anywhere on this decreasing part of the curve, then you will either get what's called monotonic dampening or damped oscillation. The system will come to an equilibrium. If, however, they cross on the left-hand side where the prey isocline is increasing, you'll get cycles. This has been used to explain how cycles happen in predator-prey systems, this model. And it's called the paradox of enrichment model because as you increase the carrying capacity of the predator, you'll go from a region where the predator can't persist, there's not enough energy in the system, to a region where you get monotonic dampening, to these damped oscillations, and eventually this limit cycle, this persistent limit cycle. But now, as you can see in this graph, I'm going to add more prey at a time, right? So what happens when we allow the predator to feed on 2 prey at a time? Well, the first thing is it can persist for longer with less energy in the system, lower carrying capacity. This dark region starts lower, right? But we still undergo the same type of destabilization, you might refer. Like as you add more energy in the system, it still goes from the stable equilibrium, eventually hitting a limit cycle, and you get this destabilization dynamics. But as you increase prey at a time even more, what happens is that the isocline, this prey isocline, flattens out at low prey abundances. What emerges is a new region of linearity associated with the linearity of the function response. And what happens is that we no longer get limit cycles. That is, we've in some sense explained the persistence of predator-prey interactions, because if you had limit cycles, there's a really high chance that either the predator or the prey will go extinct when it goes to really low population sizes. This in some sense provides us with a resolution to the paradox of enrichment. However, we also get alternative stable states, a region of bistability, where two possible outcomes are possible, just like in kelp forests. I'll show you that in a slightly different way. So if we were to start, or if we were to allow the predator to feed on, let's say 10 prey at a time, and you were to start the population, both populations, at a point relatively close to this intersection point, well, then you'd get these damped oscillations and you'd get coexistence through time at a stable equilibrium. However, if you start at a region a little further out, then you'll get entwined in a limit cycle. That is, we have a limit cycle and a stable equilibrium coexisting at the same time. As a consequence of that, what can happen are two things. We can get extremely long transient dynamics. We can get cycles that persist for many, 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 many generations that then suddenly die out without any change in the system. Likewise, if we add just a little bit of noise to our system, some stochasticity, what can happen is that we can be in this limit cycle period regime for a long time, and then all of a sudden it goes into just a little noise around a stable equilibrium, and then all of a sudden it goes back to a limit cycle. Far more complex dynamics than we would expect to see under a simple linear type 1. Right? These come from linearity. These complex dynamics come from a linear system, approximately linear system, but we wouldn't have expected them had we not introduced this additional biological reality that predators can feed on multiple prey at a time. The cool thing is that we actually see these types of dynamics in lab experiments, unexplained so far. There was a paper published 5 years ago in Nature where they see periods over these long time series, hundreds of days of cycling, and then all of a sudden no cycles, and then all of a sudden goes back to cycles in a chemostat, perfectly controlled system. So we've these same types of dynamics that we see in the simulations, we've also seen in empirical systems. I'm going to have to skip that slide too, because I want to have at least 1 minute for questions. So what's the lesson from this last section? So besides the fact that we can get really complex dynamics from linear species interactions, right? That's the first lesson. The other lesson is that mathematicians too can't ignore the biology. That assuming a type 1 function response can get you a certain distance, right? But that those approximations are only locally relevant, only locally realistic. If you perturb the system far enough away from that local equilibrium, it will go to that outside limit cycle, and your local approximation will no longer be appropriate. So biologists, the empiricists, have been correct. Systems are fundamentally linear, fundamentally nonlinear. But mathematicians have also been correct that they can be really well approximated within some bounds, some amount of variation, such that we can actually make predictions about complex systems with linear models. So, the heart of what I wanted to talk about today is that across all these levels of ecology that the lab studies, what I've come to realize is that we as ecologists need to be more careful about what our motivations actually are. Are we trying to describe data? Are we trying to predict some future state? Or are we trying to understand and explain biology? Because of the limitations of our experiments, because of the limitations of statistics, we have to make a choice between those three things. Because all our models are approximations in reality. Yes, if we had infinite data, if we had unbiased statistical methods, all models would converge on the truth. But because we are in the real world, we never can achieve that. We as scientists have to make the choice between explaining, describing, and predicting when we try to use models, not even formal ones, informal ones, to make inferences, predictions, and learn from the natural world around us. And I should better stop there because we've run out of time. So thank you. I guess I'll take a question. Francis. Yes. And that's why I feel like this is a fundamental issue for ecology. We've talked about this before. We are at the scale of our organisms. We can't we can't create experiments that have such large sample sizes that we can avoid these statistical problems. It's impossible. That's why I don't think this is an issue we can overcome. We are always going to be biased in our inferences. Yeah. But we can do really well at prediction. We just can't understand. We will be able to predict well, but for the wrong reasons. And as long as we're okay not knowing why we're predicting things well, we'll be okay. There's a fundamental imbalance. No, what's the right word? I can't think of the word. You can't get the trifecta. You can't explain, predict, and understand and describe all at the same time. You have to make a choice. One more quick question, and I'll let you guys go. This applies to BIC, to AICC. This is, yeah, I didn't dwell on that, but it's all methods. Even the Fisher information criterion, that takes into account a different form of complexity, but it's also biased. Yes, but in order to apply that to an empirical system, you need to know what the truth is to simulate it. Yeah. But applying it to a real data set, you don't know the truth. This shows that if you take realistic experiments, you will be biased. Yes. Yes. Correct. Oh, take the 500 one and subsample it. Yes, and you would have the same problem. Yes, you'll have the same problem. If you take a completely empirical approach and just treat that 500 as the truth and downsample, yeah. I should let you go. Sorry for going over. Thank you, everybody. And thank you for sitting over here. That was a lot easier.